My name is Bhuvanesh Raju. I'm a third year PhD student at the Department of Anatomy and Cell Biology, pursuing my research under the supervision of Dr. Vikram Guja. Today, I'm presenting my research work from our lab titled Exploring Differential Gene Expression in Nasal Mucus-Derived Small Extracellular Vesicle, a potential biomarker for respiratory health. Our research focuses on studying olfactory dysfunction associated with chronic nasal inflammation, where patient loses their sense of smell. It is important to understand the underlying mechanism, and because if one loses their sense of smell, they lose the entire perception of other things, such as sense of food taste, detection of harmful gases, and detection of toxins. Until today, there are no proper molecular diagnostic kit to detect the loss of smell. Our research is trying to address those gaps by using small extracellular vesicles as a tool. EVs are like small cargo packs released by our cells during normal cellular process. These cargo packs are smaller than our cells and they contain DNA, RNA, and protein within them, and they are used by these cells to communicate with each other. In our study, we collected these mucus from healthy individuals by placing a sponge on the olfactory cleft, which is the region near to the olfactory bulb, which is responsible for the sense of smell. The mucus soap sponges were collected and the small extracellular vesicles were extracted using precipitation and size exclusion method, and they were characterized based on particle size and movement using nanosite tracking analysis alongside with the Western blot. The RNA was extracted from these small extracellular vesicles and bulk RNA sequencing were performed. The nasal tissue was also collected simultaneously from the same patients and single cell sequencing was also performed. The RNA sequencing data was then compared with the single cell sequencing data to determine the molecular markers associated with the nasal inflammation as well as with the olfactory dysfunction. From our results, we have found there are some specific mRNA transcripts that were significantly upregulated, such as UGT2A and ermine. These genes are primarily responsible for metabolizing odors and are associated with the sense of smell. After comparing the single cell data with the RNA sequencing data, we have found that these transcripts are primarily expressed in the nasal cell, suggesting that they might be a potential biomarker for detecting the olfactory loss during chronic nasal inflammation. For our future studies, we are planning to uh, find how these mRNA transcripts play a crucial role in other respiratory-based diseases, and we will be validating our findings using the patient sample. Thank you very much for listening.